Hello friends and the students of medicine and physiology. A new video, a new concept, and the concept is of membrane excitation. We have discussed this in some of the previous videos, but we have touched it peripherally. Now let's discuss a core concept of membrane excitation, and why it needs an elaborate discussion is because I often see that the students have lots of misconceptions and lots of erroneous concepts related to this. So therefore, let's see what is meant by membrane excitation, what is meant by excitable cells, and what are the various possible ways in which a membrane can excite. So first things first, the term membrane potential needs to be understood. First point, because I need to develop this topic from the very basic, let's build on this concept. What is meant by membrane potential? Membrane potential is a term used for the potential on the inner aspect of the membrane, inside of the membrane. So let this be very clear that whatever membrane potential we discuss from this point onward, we will say resting membrane potential, we will say action potential, we will say depolarization or repolarization of the membrane. All these are essentially the potentials which are on the inside of the membrane. Remember, an electrode placed just inside the membrane and the potentials are being recorded. So all these potentials, whatever we refer to, is the potential on the inside of the membrane. So that's a first real thing that needs to be understood, the membrane potential. Next point is, in this context, we can divide the cells into two categories. Some cells are called as excitable cells and the others can be called as non-excitable cells. So when we say a cell is excitable, what do we mean? We mean that this membrane potential, which is a negative potential just inside of the membrane, this will become less and less negative and in fact will turn into a positive potential. That would be called as membrane excitation. The inside of the membrane becoming less and less negative and turning into positive potential would be referred to as the excitation of the membrane. So two things. One, when we say a cell is exciting, in reality its membrane is exciting. It's the membrane excitation that is happening. And the second point is, membrane excitation means inside of the membrane is becoming less and less negative and eventually becoming positive. Right. So excitable cells, nerve. Why do we need nerve to be excitable? Why do we want the nerve membranes to excite? It's a signal. When the membrane excites for a nerve, it's a signaling. All parts of the membrane will excite one by one, and that is how a signal will be transmitted through the nerve. So nerve excitation is a signal transmission through another to another nerve or to a muscle and or a gland. Muscle is an excitable cell. Skeletal, cardiac, smooth, all three types, excitable cells. And what is that required for? Muscles are required to contract, and for that they have to first get that signal for the sake of contraction, and that signal is in the form of membrane excitation. Nerve will send that excitation or that signal to the muscle. The muscle membranes, the cell membranes of the muscle will excite, and then as a result they will contract. So for the muscle to contract, its membrane has to first excite, and therefore muscle is an excitable cell. There are other cells like receptor cells. There are various receptor cells in the body. we can also call them as excitable cells in the sense that when you apply a particular stimulus to that receptor, its membrane potential becomes less negative. So membrane has begun to excite, becoming less negative. Let's take a few very peculiar examples for our discussion. Hair cell in the cochlear and the vestibular apparatus. It's an excitable cell. Its membrane is going to excite, and it is going to be a peculiar way of membrane excitation, as we will discuss. Or pancreatic beta cell for the secretion of insulin. Its membrane is going to excite, and we will see how that membrane excitation occurs. The non-excitable cell, on the other hand, its membrane does not ever excite. I mean, red cell, RBC. If inside of the red cell membrane is negative, let's say -10 to -20 millivolts, it's going to remain that way for the entire lifespan of the RBC. It will not excite. It will remain the same throughout its lifespan. So it's a non-excitable membrane, and the cell is non-excitable. That was the first point that I wanted to elaborate on. When we say membrane potential, it is on the inside of the membrane that we are referring to, and there are two types of cells. The excitable cells means their membranes are going to excite, and membrane excitation means inside of the membrane will become less negative and will eventually become positive temporarily. So let's have a few more points related to this. Let's say here is a typical cell, diagrammatic representation. Inside of the membrane is -70 millivolts. All right, it's a -70. Now this, when we refer to this as a -70 membrane potential, outside is taken to be 0. Outside of the cell is taken as 0, then inside of the membrane is -70. With respect to exterior is taken to be 0. Okay, reason is, for any cell, there are excess anions inside the cell. There are excess anions inside the cell relative to the ECF, relative to the exterior. Inside of any cell has excess anions. The proteins, the phosphate, they cannot leak out of the cell, they cannot take part in the equilibrium process. So there are excess anions, and therefore the negative charges line up on the inside of the membrane, creating this sort of membrane potential. And as I mentioned, the membrane potential varies from cell to cell. It could be -8 to -10 or -20 for a cell like RBC or epithelial cell, and it could be -35, -45, -70 millivolts in the case of nerve, -90 in the case of skeletal muscle. The value will differ from cell to cell. Whether it is excitable cell or non-excitable cell, value will be negative. It will differ from cell to cell, but it will be negative on the inside aspect. Excess anions inside the cell, so negative charges lined up, and when we say -70 millivolts, outside is taken to be 0, inside is negative to that with respect to the exterior. All right. Now, in the case of excitable cells, we use the term resting membrane potential. Resting membrane potential is one term used in the context of the excitable cells. Why? Because when they are not stimulated, their membranes are resting at, let's say, -70 millivolts. Inside of the membrane has a voltage of -70 millivolts. It's a resting membrane. And at other times, the membrane excites. That means inside of the membrane becomes positive. Now this excitation of the membrane means inside of the membrane turning positive temporarily, and then again going back to negativity, that's called as action membrane potential. Drop the word membrane, we call it action potential, as if the membrane is in action. So excitation of the membrane commonly called as action potential. Now, in the context, I want you to understand one more terminology. Look, this excess negative charges which are lining just inside of the membrane with respect to exterior taken as 0, this can also be called as polarized state of the cell. This is a polarized cell, or this is a polarized state of the cell. Inside is minus compared to exterior as taken as 0. Now, of course, there are positive charges in the ECF. So that also shouldn't be, that should be kept in mind. So this is a polarized state of the cell. When this polarity gets disturbed and positive charges come in, or inside of the membrane becomes positive, it is called as depolarization. So the membrane was in a polarized state, the cell was in a polarized state, or its membrane was in polarized state, and now that polarity got disturbed and inside of the membrane has become positive. So disturbed polarity, it's called as depolarization. And then again the membrane recovers from this excitation, and inside of the membrane again goes back to its negativity. It will be called as repolarization. Simple. Disturbed membrane polarity, depolarization. So inside became positive. And then again it went to its original negative potential of minus 70 millivolts. It would be called as repolarization. So remember, basically action potential, the membrane excitation, has these two phases, depolarization and repolarization. But in reality, you should remember, for the sake of concept, you should remember like this. Membrane excitation means depolarization. It is the depolarization that travels through the nerve like a traveling wave, like an impulse. And when the membrane recovers from that excitation, it's called as repolarization. Yeah. So quickly, once again, polarized cell, a resting cell, it was resting, its membrane was resting. When that polarity got disturbed and inside became positive, we call it depolarization. Inside again became negative, its original potential, its original value. So membrane has recovered from excitation, it's called as the repolarization. That polarity got restored again. One more point. When this polarity becomes excessive, originally the membrane was supposed to be at minus 70 millivolts, and suppose some negative charges come into the cell, membrane can become excessively negative, or some excess positive charges leave the cell, membrane becomes excessively negative, minus 90 millivolts instead of minus 70, that would be called as hyperpolarization. So that polarity of inside being negative, it became excess negativity of the membrane on the inside. That's called as hyperpolarization. And remember quickly that the hyperpolarized cell is a temporarily inhibited cell, means it will be difficult to excite the cell if it is hyperpolarized, if its membrane is in a hyperpolarized state. So excess negativity called as hyperpolarization. These are the terms that should be understood clearly in this context. If we look at a non-excitable cell, as I mentioned, if it's a negative potential on the inside, it will remain so throughout the lifespan of that cell. The membrane will not excite. So minus 10 will remain minus 10 for the entire lifetime of that cell. Alright, so that's the excitable cell, that's the membrane excitation, and we can call depolarization actually is the membrane excitation. and the various values. Now coming to the crux of the matter. What are the various ways in which the membrane can excite? What are the various ways in which the membrane can depolarize? Let's look at this particular aspect, which is actually the reason for this video. Let's say here is a typical cell with negative charges lined up on the inside. And as I said, membrane excitation means inside turning positive. Well, let me halt here. Inside becoming less and less negative from -70 to let's -50. That also is a depolarization. Moving towards positivity. It's called as depolarization. And a membrane completely depolarizes or completely excites when it insides becomes positive. So in the case of receptor cells, membrane may not completely become positive. It can move from -70 to -60 or 50, -50. That is also the membrane excitation for the receptor. In the context of a receptor, we can say moving 20 millivolts towards positivity from -70 to -50. It's a 20 millivolts depolarization. That also is a membrane excitation. Well, here we are talking about, let's say, a nerve cell. Inside completely turns positive. A nerve cell or a muscle cell. That's membrane excitation. And what are the ways in which this can happen? That is the video is all about. First, look, the cations which are present just outside the membrane can flow in. So, which is the cation with high concentration in the ECF? It's the sodium. 141 milliequivalents per liter on the outside, 14 milliequivalents per liter on the inside. So it has got a good concentration gradient to come from outside to inside. Diffusion can occur through its channels. And therefore, first way of membrane excitation is influx of sodium. Through the voltage gated sodium channels, sodium will come in. Membrane has been stimulated, so these channels will open. Sodium will come in. So positive charges are brought in. These positive charges lining up on the inside of the membrane has excited. Where do you see this commonly? Of course, nerve. The nerve membrane excites due to the influx of sodium. Simple. This is the most commonly known way of membrane excitation. We generally talk about entry of sodium resulting in depolarization. That's in the case of nerve. Most common form of depolarization. Most common way of membrane excitation. Second, which other cation can do this? Calcium. Yes, in some cases, calcium influx can also result in depolarization of the membrane. We all know that the calcium is very less in the ICF. You know, cytosol cannot have a high concentration of calcium. So calcium is relatively very high on the outside and cytosolic calcium concentration is very low. Intracellular calcium is kept very low, and therefore calcium also has got a nice concentration gradient. So influx of calcium. This is even easier because calcium is a divalent cation. So whenever calcium comes in, it brings in double the number of positive charges compared to sodium. And therefore, calcium influx can also cause excitation of the membrane. Where do we see this? Well, heart, SA nodal cells, they depolarize due to various ions actually, but the major part of their depolarization is because of the influx of calcium. Remember, calcium plays a very important role in their depolarization and excitation of the heart cells, particularly SA node, but even Purkinje fibers. Calcium is very important. Yes, so the calcium is also important for the contraction of the heart muscle cells. So it causes this electrical excitation of the membranes and then goes on to cause contraction as well. The same calcium is used for the contractility in the heart muscle. We will see that in some other video. Third, any other cation? Potassium. Yes, this is what I need you to understand. I want to draw your attention to the potassium. Whether potassium can bring about membrane excitation, membrane depolarization. Possible? Look, why am I asking this question? The reason is, we have learnt in the case of nerve that first the sodium comes in, so membrane excites, and then the potassium goes out. So let's just draw it here. You know, potassium is high in the intracellular fluid, let's say 145 milliequivalents per liter, and it is low on the outside, you know, about 4 milliequivalents per liter. So potassium's concentration gradient is favoring the potassium to go out, isn't it? High to low concentration, inside to outside. And when the positive charges go out, they leave behind the excess negative charges. That is what is repolarization. So we have all learnt that the potassium goes out and causes the repolarization of the membrane, the recovery from excitation. But my point is, can potassium cause membrane excitation? Can it cause membrane depolarization? The answer is yes. So, potassium influx can result in depolarization. So, first question that you might ask is, how can the potassium influx happen? Because potassium is high on the inside and low on the outside, so potassium will always go out by its concentration gradient. How can the potassium come in? Now, this is all correct, but there is an exception in the body, and that exception in the body is hair cell in the cochlea. Hair cell in the cochlea and in the vestibular apparatus. It's a flask-shaped cell. It's involved in the process of hearing. It's involved in the process of equilibrium of the body. The hair cell is bathed in a fluid called as endolymph. Endolymph is surrounding the hair cell. And what is the speciality of endolymph? What is the uniqueness of endolymph? Endolymph is an ECF. Endolymph is an extracellular fluid. So it has got all the features of extracellular fluid, all the concentrations of extracellular fluid, and excess potassium ions. Excess potassium ions over and above the ECF composition. So the endolymph, which is surrounding the hair cell in the cochlea and vestibular apparatus, it has got ECF composition, extracellular fluid composition, plus over and above that composition there are excess potassium ions. That means this potassium, which is excess on the outside of the cell, which is not the case with any other cell in the body. For all other cells, outside the cell potassium is low. Here, an exception, the endolymph has got excess potassium ions. Which means what? Which means here the potassium can enter the hair cell. Influx of potassium is possible. So whenever there is sound, there would be bending of these hair, the stereocilia at the top of the hair cell, and that will open the potassium channels which are at the base of the stereocilia. Look, at the top of the hair cell or at the base of these hair, base of the stereocilia, there are these potassium channels. And during the process of equilibrium, head rotation, or during the sound when it enters the inner ear in the cochlea, there will be bending of these stereocilia, and that will open the potassium channels. Potassium influx will happen. Positive charges have entered, and therefore there will be excitation, depolarization of the hair cell membrane. So this hair cell was also negatively charged to begin with, but influx of potassium has caused depolarization of the hair cell. And this depolarization actually opens the voltage gated calcium channels in the hair cell membrane. So, because it's a voltage change, of course, therefore voltage gated calcium channels will sense it and they will open. Voltage gated calcium channels open, the calcium influx happens, and that results in exocytosis of the glutamate. Glutamate is a neurotransmitter which is present inside these vesicles. So, neurotransmitter will be released and 8th cranial nerve, which is synapsing with the sides and bases of the hair cell, there will be impulse generation or change in the impulse traffic in the 8th cranial nerve fibers. But the point was, can potassium cause excitation? Yes. Exceptional case, potassium influx could happen. And why exceptional? Because at other places, as I mentioned just now, that the potassium is high on the inside and less on the outside. So potassium is has got a force to move out from inside to outside. But here is a case where potassium influx can cause depolarization and membrane excitation. Is there any other way? Yes. Now that's the most interesting part coming up now. Look, there is a cell and its membrane is resting, negative potential on the inside. And we have said that inside of the membrane turning positive is membrane excitation, depolarization. Now, it can happen either by influx of cations, positive charges from ECF, they can enter. We saw sodium, most common. We saw calcium, and even in the exceptional case, potassium as well. There is one more way. Let's say inside of the cell, at the core of the cell, there are anions. We know that there are excess anions in the ICF, the proteins, the phosphate. These are anions which cannot leak out of the cell. So, anions, excess anions inside the cell. Surrounding those anions are the potassium ions. We've already discussed this, that the potassium is high in the intracellular fluid. So, it is near the core of the cell. Is it possible that this potassium, which is at the core of the cell, just comes near the membrane? From the center of the cell, it just moves near the membrane. Is that possible? Yes, it is possible, and even this can result in membrane depolarization, membrane excitation. The membrane which was negatively charged on the inside, just on the inside, now these positive charges in the form of potassium, which was at the core of the cell, they move toward the membrane. Now basically, why does it move toward the membrane? It's not going to, the original purpose of that was to go out of the cell. Yes, that was the force for the movement of potassium. This intracellular potassium was being forced to leave the cell, to go out of the cell through its channels in the membrane. But if the channels are closed in time, what will happen? The potassium has started moving to go out of the cell, but channel got closed. So potassium could not go out. It just comes near the membrane and stays there. Membrane has become positive. But by which ion? The intracellular cation this time. So potassium at the core of the cell, it had the force to go out. It started going out, but its channel was blocked. Potassium could not go out. Channel was closed. So it just came and stays inside the membrane. Membrane got depolarized again. The membrane got depolarized. Right. So, right? This also is one more way of membrane excitation. And why I took also this part into consideration, because the pancreatic beta cell is known to excite in this manner. In the case of release of insulin, membrane excites, the cell excites in this manner. Let's see how. Let's say here is a pancreatic beta cell. It's going to release the insulin by exocytosis. So, we all know that insulin is released in response to increase in the blood glucose. We eat glucose, we eat foodstuffs, and there is increased blood glucose. Now this glucose in the blood, in the plasma, it will enter the cell, pancreatic beta cell. Now this glucose which has entered the cell, this is going to cause release of insulin. Yeah, we know that already, that the insulin release occurs in response to rise in the plasma glucose, blood glucose. Let's see how. Blood glucose increased after the meals. This will undergo glycolysis. All right. Glycolysis will generate the ATPs. Correct? Now, in the membrane of this pancreatic beta cells, there is a peculiar channel called as ATP sensitive potassium channel. And when the ATPs are generated inside the cell, sensing those ATPs, this ATP sensitive potassium channel will close. Well, ATP sensitive potassium channel closes because the ATPs have been generated. They are sensed by the channel will close. Now, because this channel has closed, the intracellular potassium which was expected to go out through the channel will now not be able to go through the channel. It won't be able to exit. No efflux of potassium possible. So, intracellular potassium which was going to be driven out of the cell now cannot go out. That means potassium starts accumulating near the membrane. As I mentioned just now, from the core of the cell, the potassium just comes near the membrane. It starts accumulating inside the membrane. Well, that's it. Membrane excitation has happened. The cell has excited. Now, this membrane excitation will open the voltage gated calcium channels. It's a voltage change for the membrane. So, voltage gated calcium channels will open. Calcium will enter. Calcium influx will happen. And this calcium influx will result in the insulin to be released by exocytosis. Insulin was stored in these vesicles. Now, this insulin will be released by exocytosis. That is how insulin is secreted by the pancreatic beta cell. So, let's go in the reverse. The beta cell in the islets of Langerhans, it has insulin in the vesicles. Now, these vesicles have to release the insulin by exocytosis, and for that to occur, the vesicular membrane has to fuse with the cell membrane, and that is possible by the help of calcium. But calcium is low intracellularly, therefore calcium has to be brought in. Calcium is going to come in by through the voltage gated calcium channels. So, they need to be opened. That means the membrane has to excite, and membrane excitation could occur in this manner by the intracellular cation, that's potassium. Just from the core of the cell, it comes near the membrane excited. So, instead of having two cations coming one after the other like sodium, then calcium, and then causing insulin to release, it's the intracellular potassium. Cell is rich in potassium, it just moves near the membrane. Membrane excites from inside, and because of the intracellular cation I am saying, and therefore that opens the calcium channels, insulin will be eventually released. So, these are the various ways in which membrane excitation can happen. In summary, inside of the membrane turning positive, well, let me just add here, depolarization can also be partial. I mean, membrane does not have to become completely positive, but moving from just -70 to -50 also is moving towards positivity, becoming less negative. That also is disturbed polarity, or it can also be called as depolarization. That's the point needs to be added here. And these are the 4 ways in which the membranes can excite